I have to tell you right now that this title is far from the kind of title I would normally use. It's a Foreste kind of title. Foreste has been a huge helper in what I did. And let me begin by trying to remind you of a few of the points Foreste and the other speakers that came before me said that will be important in what we're talking about now. The main points that they made were no two cancers are alike. Cancer is a highly heterogeneous disease. Every cancer type has multiple subtypes. They evolve over time. They become more aggressive, as we saw in Shirley and Vanessa's tumors, and they get better at evading the immune system and spreading around the body. So a tumor starts as a single corrupted cell with whatever mutations it has at that time, and then it changes over time and becomes more complicated. And having a good way of visualizing how this process is going on, especially in the context of other tumors that we have information about, would be very useful, we think, and in understanding specific tumor evolution, and in the context of other tumors of different types, understanding possibly when changing treatment strategy should occur right away and how to personalize somebody's therapy. The important thing that does happen, and Foreste mentioned it, is sometimes due to things that happen to the patient, the behavior of a tumor may change and eventually the tumor might behave less like the tumor it was originally diagnosed at as Foreste suggested, and might go from being an ovarian tumor to some kind of aggressive sarcoma. If we can identify that this is happening, right, it might, if the appropriate drugs are available, signal a recommended change treatment strategy. Our hope, and the thing that I will attempt to convince you we can see, is that certainly in multi-omics data, but simply in the pattern of changes in RNA expression levels for many genes, the important changes in tumor behavior may become evident. And you should notice that we've seen many, many times that while the tendency now is to focus on a small number of genes that people think are important, it can turn out that the changes in the pattern of expression levels over many genes are more significant than what's going on. So the technology that we use to try to talk about this behavior is something called dynamic quantum mapping, or DQM. And I'm going to show you results for both the Foreste and Vanessa's genes at the same time, their tumors, plus other tumors that Foreste supplied me with, all of which was produced on the ImmunoID Next platform that Personalis provides. One thing I have to talk about, which is important, is why am I here? I'm not a biologist. I'm not a doctor. I have only a growing but small knowledge of biology and medicine. However, my training is as a particle physicist. That's how I started out 1,000 years ago. And what happened, my expertise is really in the area of quantum mechanics, quantum field theory, non-perturbative computational methods, and data mining, okay? I spent the last 40 years of my professional life at one place, the Stanford Linear Accelerator Center, or the SLAC National Accelerator Laboratory, as it's now called. And I'm the co-inventor and developer of dynamic quantum mapping. Analyses that are often used, and you heard them talked about, to find cancer-driven genes and pathways using transcriptomics data is one, differential gene expression analysis. You try to identify important genes by saying their expression levels are elevated over what they are in normal tissues. The only problem with this is we rarely have samples of truly normal tissue. For example, in the ovarian cancer case, it's almost impossible to find truly normal ovarian tissue. Who would want to give up and be biopsied if they're not sick? So the tissue you get is at best tumor adjacent, and and since we don't know the normal expression level of a given gene, it's hard to know which ones are correctly being identified as highly enhanced or suppressed. There's gene enrichment analysis that looks for a set of differentially expressed genes or a predefined set of genes. Again, that's it's based on literature searches and on knowledge based in the community, but it is not completely agnostic about what genes and what large sets of genes might be contributing to what's going on. Network analysis, of course, plays a role but we don't have that information for lots of these cancers. Survival analysis is great once you have clustered the things, once you have subsets of samples that you know are important to understand. It lets you say, it certainly lets you see if something you've done to treat the patient has improved their survival. Then there's co-expression analysis and pathway analysis, and all of which play important roles, but pathway analysis in particular here rarely it's based on sets of genes that are supposed to operate in a particular way, and those pathways are determined in a way which is independent of the specific data you're looking at. And so it would be nice if we could actually understand those effects better using only the data that's going into looking at the treatment. Dynamic quantum mapping is a way of doing something that's like clustering, but goes far beyond it. It is capable of identifying subsets of the data which are more similar to one another. It can do it in arbitrary dimension, so you don't have to do hard reduction with using PCA. The way it works is let's use something like PCA to map the data out in 2 or 3 dimensions for an example. And what DQM does, it creates a terrain map, a function basically, that when you map it, and the lines on this figure are equal height lines, okay, those elevations of those regions are determined by how many data points are in a given region locally. So regions where there's more data close to one another, those are regions of higher similarity for that data, as you see here. I mean, this is real data. This is not made up. This is data from a very famous old article on data mining. They actually end up separating the regions, not perfectly, as you see here, but the points that lie in a lower region where there's more data, in fact, those points are the ones that correspond to a single type of, in this case, crab. The idea is I can show you this map, as with other maps, in 2 dimensions. I could show you similar kinds of things in 3 dimensions to some degree, but then when I'm dealing with data which has 22,000 genes as features, there's no way to look at the map. But what I can do, which can be done in any number of dimensions, is I can say, well, if I take all the data that belongs to a depressed region, if I can collect that cleanly, then basically I can map out that region of the locations of the depressed regions, and the signal that things are now all together in one place will give me an understanding of what's happening in higher dimensions. The trick that I use, because of my background, is the goal is to move the various data points downhill in their depression until they all arrive at the same location, the bottom of the depression. Now, obviously, some points will be misidentified, as we see here. They will move downhill into the wrong region. But the purity for most things, some of that can be experimental error. Some of that can be that the points were originally misidentified. And basically, you might think that the right way to do this is mathematically, for the mathematicians in the audience, gradient descent, but it's well known that that gives rise in high dimension to all kinds of problems. A method which avoids those problems is to treat these data points as quantum mechanical particles moving in a potential function defined by the terrain map, and let the points move under quantum evolution, which is different from gradient descent. Let them move together and collect. What we do is we will show what happens, the pictures, the animations you'll see, what happens when you wait a little time under quantum evolution, the points move, you get another picture, points move, you get another picture. In the end, you have an animation. That animation is actually a visualization of the complex mathematical calculations that are going on and moving these points together. The neat thing about this trick is it works in any number of dimensions. So here is the data that Ferenczi supplied. It is the data cohort supplied by Stanford. You see the evolution of the points. This is being done for all 22,955 genes provided by the Personalis platform. Since there are only 40 points in this sample, it's being done in 25 PCA dimensions. And the only reason for that reduction is because there's nothing changes if I go to 40 dimensions. It's the same picture that you will see. The important thing is, two things are going on. These are PBMC points. These are immune system cells that were in contact with the tumors. Over here are the tumors that are related to ovarian cancer. Coming in from here, as a big surprise, is a pancreatic tumor that was in the data set. Down here are some control samples. The red points that we see are, so here is a red point, that's a Shirley PBMC point. Here are two Shirley points, which were provided originally. Here is a third one, okay, which does not cluster with anything. That is the less, these have evolved over time. So these points originally, I forgot to say, unlike the other clustering maps you've seen, the distance between points here is meaningful, especially at the end of the evolution. In the beginning, you're only seeing the first three PCA dimensions. But as things come together, they're doing it in all 25 dimensions. And I could show you plot after plot to prove that. So basically, you see that first, all of the samples that are ovarian tumors are fallopian tube control samples, are primaries or relapses, are all coming together in one cluster. This pancreatic tumor comes in at the end. The length of time it takes to arrive at the cluster is a measure of how far away it started from the cluster. So literally, you can see that these pancreatic controls, this pancreatic tumor has nothing to do with these, but one of these is coming in. And that was a great surprise to me, and I'll have something to say about that later. But you can see that this is showing you what's going on. This is exactly the same thing, except we're listing the members of each of the clusters. So the big cluster, the one which is the one we call cluster one, that was everything you saw going into that circle. And as you can see, it contained the fallopian tubes control samples. It contained direct ovarian primaries and relapse. That was the dark blue. This was the lighter blue. And basically, two of Shirley's tumors. There's a lymph node tumor from a lung sample, and there's the pancreatic tumor. The yellow stuff down here, that's cluster 2, and that's normal skin control samples. Here's the other Shirley sample. These all don't coalesce with anybody. And basically, this is the information that's contained in that slide. Sorry, I was just looking to see if there was anything I could answer at the moment. Here's the longitudinal transcriptomics. So now I'm showing you exactly the same slide, but I've rendered the samples that were not either Shirley's sample or Vanessa's sample or the pancreatic samples, that one of which comes in here. From this picture, you absolutely see, right, that the Shirley samples evolved in time. These two were earlier, and they, in fact, are more like one another. This is quite different, and this is the immune cell related sample. So there's some clustering of Shirley's sample, and then basically we see Vanessa's samples never move. There's the desmoid tumor, and they are not like the other samples in this thing at all. They're far away from one another, and they are not clustering. They're all individual. We saw that Vanessa's disease had undergone major changes, and it is reflected in the RNA sequence data. Again, this is the nature of the samples. The coloring was what you saw, and the hypothesis that the RNA-seq data will capture the evolution of the tumor over time. It's very clearly seen, and yet it doesn't lose the relationship of things to one another, as we saw that we can see the point in time where major changes have occurred in Shirley's sample and where things have remained somewhat similar to one another in the early thing. So this is what we're able to do with this small amount of data. We can see what's going on. We can see the evolution of the tumor, and we can try to see what's happening. It would be more helpful, right, if we could place this tumor in the context of everything we know, a huge data set of other tumors. And so now I want to show you something that we tried, and it's a challenge, and unfortunately, I'm going to invite people to try to join us in an exercise at the end of this thing. The idea was to look at the TCGA, the Cancer Genome Atlas data set, and combine it with Shirley's samples and with Vanessa's samples. So namely, here we're looking at the primary tumors and a whole bunch of other tumors from TCGA. There are 15,000 of these on this plot. You see that DQM is being applied to this, so this is already what you could call the clustered thing, showing different tumor data sets. And basically what you see here, again, the Shirley tumors, not including the PCBM, and Vanessa's tumor set. But what I didn't tell you about is over here. This is from TCGA, and the blue is the ovarian tumors in TCGA. And this little bit of cyan that you see here are the pancreatic tumors in TCGA. And what you see here is that the ovarian samples in TCGA break up into many clusters. That is actually, here's now I'm hiding everybody except TCGA's ovarian samples, TCGA's pancreatic samples. And what we're seeing here is what we've heard constantly, that ovarian cancer is highly variable. There are many different kinds of ovarian tumors, and they are clustering differently. And then we see, in fact, that we ask the question, why are the pancreatic tumors managing to move in to Shirley's tumor? And we see in all of TCGA, in fact, the pancreatic tumors cluster a short distance away, from a subset of the ovarian tumors. So presumably, it's not a surprise that some pancreatic tumor samples will cluster with a subset of ovarian tumor samples. Okay. But what's the problem? The problem is that we look at the Shirley data and the Vanessa data in the context of all of TCGA, and they don't move. They're not aligning with the other tumors. They're being misidentified as having nothing to do with ovarian tumors, nothing to do with pancreatic tumors, and they can't be true. So what's happening here is the Personalis data has a systematic effect, which is shifting it away from the CCGA data. So while I'm able to take all of this stuff and analyze it, here's the PBMC data you see, it comes back. I'm able to analyze this completely in terms of the TCGA data. There is a systematic effect which is making it useless to try to combine this data. This is a huge problem because it means the bulk of all of this data that could be useful, after all, if we saw these tumors and saw how they changed and were able to place them in the context of TCGA, we would be able to say at this point in time, this tumor has become more like a sarcoma and less like an ovarian tumor. And in that case, that's a clean signal. You should be thinking hard about how to change the treatment strategy because you're being told that at the molecular level, the cancer has changed its characteristic in a way that is dramatic, that it's behaving more like a different tumor than what we thought it was when we started out. So to summarize, what did I want to say? We know tumors change in time. We believe understanding how they're changing time will affect the decisions about how they should be treated. These changes can be captured and mapped using DQM as an example by the changes in RNA expression levels for the biopsy tumor. And the DQM tool allows us to simultaneously visualize the changes of the tumor in relation to other tumors in high dimension. It can cluster all of the data without hard dimensional reduction to make these correspondences easier to understand. It's easy once you've done this to identify a smaller set of genes, order typically 50 to 100, which is a lot less than 23,000 genes. And they have to be chosen in such a way that the pattern that you see in the DQM analysis is the same. Not only the original picture, but in fact, the time evolution and the clusters that are formed. And once you see these different clusters, then you can go in, having reduced the number of genes, and do a gene set enrichment analysis, a network analysis, a pathway analysis with all of the data, let's say TCGA plus the new data for the specific patient. But as you've seen, I can't do this now because the Personalis data, the stuff produced by their IMMoID Next data platform, and TCGA data are incompatible. The question is, we're faced with, right, a Hobbesian choice. One thing we can do is wait for Foreshte to just get another 22,000 samples of different patients, all done on the Personalis platform, which would be really nice. It'll be really expensive, but it'll be guaranteed to be data that's compatible. Or we can try to figure out what's going wrong. And for example, by looking at the housekeeping genes, could we figure out that there's an easy rescaling that can be done and shifting of the Personalis data to make it align correctly with the TCGA data? I'm not an expert on TCGA. I know that out there among you, there are people who know a lot about that. And I would love you to get in touch with me and see if we can put together a collaboration to figure out how to do this. If we can, the ability to help fuse your patients in this RTTP process would really be greatly increased. Our software, our company, Quantum Insights, has officially gone out of business, and we've taken our proprietary software, DQM, and made it open source. A Python version of what you see above has been made available for download on GitHub. And if you go to https://DQM for dynamic quantum mapping read the docs, that will explain to you how to download the software, how to install it on either Unix or Windows or iOS, and use it for analysis. We would love to have people who have some knowledge about TCGA and what is done in preparing their data and how it might compare to Personalis. Get in touch with me and proceed to maybe move on to the next step in this thing, so we can actually use the visualization tool. Otherwise, we're just going to have to wait for more consistent data developed on a single platform to become available.
Marvin.